Good morning, everyone. Hi, my name is Ivana, and I'm a science program manager at Zuckerberg Initiative, and I'm excited to be here today and tell you more about our programs as well as funding opportunities. Can you hear me okay with the mask? Or should I maybe take it off? Okay, this should be better. So our mission at CZI is to support the science and technology that will make it possible to cure, manage, or prevent all diseases in the next 100 years. However, our 10-year plan is to accelerate biomedical science by developing new tools, technologies, and supporting open, collaborative models of research. And this is centered around our values around people, technology, collaboration, and open science. And so we aim to accelerate biomedicine through funding. We have a number of CZI science programs that I will also tell you more about, grants to scientists worldwide. We also build tools and technology to help scale science. We engage with patients and public as partners in scientific process, and we collaborate with partners and communities to enable progress across fields. And just to share some of the technology that we build for science, and it's still openly available: cell by gene, Napari, CZID, Shasta, GNFP, and FISH. And I'll tell you maybe just a little bit more about some of them because they may be of interest to some of you doing research in these domains. And so cell by gene, for example, is an open source tool for exploring single-cell transcriptomics datasets. Napari is an interactive multidimensional image viewer for Python. Shasta is the de novo genome assembler for long-read DNA sequencing, and Starfish is an open source Python package that helps with spatial transcriptomics data. On the program side, we support experiments and accelerating science. And our major programs include imaging, neurodegeneration, challenge network, open science, single cell biology, science and society, and CZ BioHub. And success for us looks like enhancing productivity through publications, preprints, software, data sets, and protocols and other resources. Disseminating those in public repositories or enabling others to do so, requests and reuse of citations and clinical applications, as well as commercial development, and collaborative contributions through leadership, co-authorship, success of students, postdocs, and acknowledgements. And I'll just go briefly into all of the programs and tell you about them. And then I can answer any additional questions if you have them after. So for neurodegeneration challenge network, we aim to attract new talent and ideas in neurodegeneration fields and create new models for collaboration, open science that will accelerate understanding of neurodegenerative diseases. Open Science program aims to empower more people to engage in research and accelerate pace and robustness and reproducibility of science. This is done through a number of projects, and I list here some of them. USS, or Essential Open Source Software for Science, is a program that just concluded a fifth cycle, and it's supporting maintenance, growth, and development and community engagement of open source software projects. This program also supports platforms that enable rapid sharing, like bioRxiv, Protocol.io, and medRxiv, to quickly disseminate and openly disseminate methods and preprints. And then building computational capacity through efforts like supporting efforts like Carpentries to facilitate access to computational training for biomedical researchers. Imaging program aims to enable researchers to visualize, measure, and analyze biological processes underlying health and disease. And this as well is done through a number of projects and Some of them are listed here that include in supporting imaging scientists to increase collaboration between biologists and technology experts in imaging core facilities and improving imaging tools, supporting technologies such as deep tissue imaging technologies, which is a part of the frontiers of imaging effort, and at the same time supporting imaging software fellows to continue development, and global bioimaging, which aims to strengthen community building and training worldwide. Science and Society is another program at CCI, which is helping build support for science by engaging patient organizations and policymakers and other key stakeholders. And two of their main projects are RARE is One and Movement for Science. RARE is One is connecting rare disease patient advocates and enabling them in their quest for finding cures. Movement for Science is building public trust in science so that support for science is commonly held value, and so that CCI efforts can cure, manage, or enable curing, managing, and preventing all diseases effectively and in a scaled manner. CZ BioHub brings together physicians, scientists, and engineers from a number of universities in the Bay Area, including UCSF, Berkeley, and Stanford, to encourage collaboration and accelerate biomedical research. And lastly, single-cell biology program, which is the one that I'm a part of, aims to understand the human body and diverse disease at the level of fundamental unit of life, single cell. And we aim to accelerate single-cell biology methods, technologies, and applications. And in turn, support generation, assembly, and use of healthy single-cell references that clarify cellular disease. And we do that through three mechanisms. One is funding, the other one is building, and the last one is partnering. With a goal to build or enable building single-cell atlases that span a variety of tissues, modalities, ancestry, and age. And in order to do that, we had launched a number of programs. Some of them already are in motion, such as seed networks and ancestry networks to enhance representation within those data sets, and then pediatrics to include data sets from patients of different age groups. On the funding side, we are well aware that a future for everyone requires diversity in terms of patients and researchers and institutions, and so we embed that in all of our programs, and including funding mechanisms. And last year we started the Ancestry Network through Ancestry RFA that aimed to address lack of representation in data generation effort, as well as improve single-cell tools and, again, work towards our goal of understanding mechanism in diseases, but in an inclusive manner. Our funding falls in these three categories, and so the first one is supporting collection of data. This is a 3-year grant program to support DCP, data collection platform, with a priority to store raw data sets. Gaining insight is the next category, and I will talk more about this one in a moment, but this is funding for data reuse and development and improvement of computational tools. And then the last one is the new 3-year grant to support BioNetworks draft atlas construction. Data Insight program is a new program. We just it's a 3-cycle program. We launched it last year, and the second one is open right now, and I'll tell you more in a moment. But it's motivated by the fact that large amounts of data are being generated, and it aims to leverage existing data sets to expand computational biology ecosystem. which will then in turn help gain meaningful insights into biology and technical progress towards subsets of analytical challenges in single-cell biology space. Application portal is open until August 25th. And like I said, all the projects must reuse existing and openly accessible data sets. Focus areas include meta-analysis of single-cell data sets, developing scalable and robust tools and methods for data analysis, problems in spatial transcriptomics, improving existing tools, standards, or increasing interoperability of existing tools, developing benchmarking frameworks for various tasks and infrastructures within this space. And I forgot to mention that we did recently announced 20 new projects that were funded through Cycle 1. The current cycle that is open and will remain open through end of August is Cycle 2. And there will be one more after that. I did mention that we also partnered with other community members and build bridges within different communities. And so we support Open Problems and Single Cell Analysis Group, which is benchmarking the various methods and approaches within single cell data analysis space. So they also organized a competition at NeurIPS, which is one of the leading machine learning conferences in 2021. And they will do that again this year in 2022, and we'll continue to support them. And this is a great way to connect machine learning and data science communities with single cell biologist community. We also collaborate with other communities and convene meetings and workshops. Since they're on the right side is a photo from a workshop that we organized in April, where we invited researchers from many different institutions worldwide. There were representatives from Australia, Eastern Asia, as well as Europe and US, to work on specific problems. And so this particular group worked on data integration, single-cell data integration problem for several days, and made a tremendous progress, and we all learned a lot. So we plan to continue hosting events like that. I did mention earlier that we also built tools and different technologies, and again, open source them. They're freely available. You can access them through our website if you're interested, or find me after, and I'm happy to talk more about them. Related to single-cell biology is CZ cell by gene tool to discover mechanism of human health, but it comes in two flavors as Gene Portal and Gene Desktop. Cell by Gene Portal is a platform to maximize impact of published single-cell data sets by making them findable and usable to as many researchers as possible. There are currently 32 million cells on this portal with 24 million primary cells, over 400 data sets organized in 72 collections, 6 modalities, and over 500 unique cell types. This is what the portal looks like, and the website is cellbygene.cziscience.com. Feel free to explore if this is your area of research. Cell by Gene Explorer is a data visualization tool. And so now you can take any of those data sets, open them in this Gene Explorer tool, and interactively explore a data set using this no-code interface, and see how patterns of gene expressions are determined by their genetic or environmental factors, in color by different metadata, such as cell types or disease, or perform differential gene expression between custom groups of cells. We're also constantly working on new features, so keep an eye on the website if this is your area of research. Outreach is another way that we stay connected with the community through movement capacity building. There are some resources on the website for anyone that may be interested, but this group partners with CZI Science program managers and colleagues and organizes training. I included there some examples of leadership training, career building, mentoring, such as on topics like diversity and inclusion, with the goal to create a network and provide added value in addition to just funding. And so all of our programs aim to create cohorts and support them through the duration of their grants. And on the note of funding opportunities, I did mention data insights, but we do have currently 3 requests for applications open. Advancing imaging through collaborative projects is one of them, and so with the goal to reduce imaging ecosystem fragmentation and accelerate speed and adoption of technologies, methods, and training resources. I already talked about single-cell biology data. Data insights, this is cycle 2, and dynamic imaging, RFA, that aims to advance technology directed at real-time visualization of biological processes. This is my team, so our teams are also very diverse, and that enables us to do things slightly differently than maybe some of the other organizations. And because we are both funding, building, and collaborating with others, we do have a large engineering team, data and statistics teams, as well as product design and science program. And with that, I would like to thank you for your attention and answer any questions if we have time or if there are any questions.
There's the mic.
Hello. Yeah. Thanks for your introduction. Yeah. And yeah. So I'm an incoming system professor, and I know there are very well-established people on the board. So do we have some specific track for the junior faculty? Thank you.
We don't have a specific track for junior faculty, but all of the programs are open to everyone, including junior faculty.
Okay, thank you.
Yes.
Hi, can you hear me?
Yes. Yes.
Thank you for our presentation. Do you actually allow cross-pollination between programs? Let's say if you want to work on the neurodegeneration challenge with single-cell approaches, and who will you contact if you want to go that way in terms of, it seems like all your teams are a little bit separated?
Yes, we do. We actually just closed RFA, request for applications, at the intersection of neurodegeneration and single-cell biology.
Okay, I wish I had known about that.
You can stay connected with us through some of those links. The science funding is a really good one. That's always up to date. But yeah, you can also follow us on Twitter or reach out.